Okay, friend.
All right. Welcome everybody to today's Crop Talk webinar series. It's the final one of 2023. So hopefully if you joined in with the previous one, you've enjoyed this series. This year we've had good topics and speakers throughout the series, and to get some good information to help you out on your farm operation, or to help if you're advising farmers. You can view the previous sessions recordings at northwest.ksu.edu website, or on the KSU Agronomy YouTube page. And there we go. And just to acknowledge everybody behind the scenes here that has made this Crop Talk series possible. Takes a team of us from the Northwest region. Got myself, Craig Dinkle, the crop production agent from the Midway District, which serves Russell and Ellsworth counties. Sandra Wick from the Post Rock District. Cody Miller with Phillips Rooks. Keith Van Skyke from Twin Creeks District, Aaron Highland from Rollins County, Emily Bingingsdorf from Thomas County, multi-county agronomist Jeannie Falk-Jones from the Sunflower District, and Kelsey Wirtz from the Golden Prairie District, and Lacey Norderman from Walnut Creek District, and Stacy Campbell from the Cottonwood District. Like I said, the handouts for this session are available. If you didn't get them, you missed that email at northwest.kstate.edu/events. And like I said previously, you can find the previous session week's Crop Talk series recordings there too. And that link will be in the chat box. And, if you have any questions throughout the talk today, you can use the Q&A box, down at the bottom of Zoom to ask a question, or you can put it in the chat box and we'll address usually most of the questions at the end of the presentation. And then if you're on YouTube, you just type it into the YouTube comment section and that will be relayed back to us through Aaron Highland that's monitoring YouTube. And if you need CCA credits, there's two ways you can get those credits. You got the QR code there that you can scan and take a picture of to get those credits, or email your name and CCA number to Jeannie Falk-Jones at ksu.edu. That's jfalkjones@ksu.edu here at the beginning, and then again at the end to receive. She'll get those CCA credits applied for you. And to introduce our last speaker for the series, we got Dr. Rachel Veenstra, also known as the Tiller Queen. She just recently took the appointment as a teacher with the Department of Agronomy, which was previously held by Dr. Donnelly. Teaches introductory to crop science courses as well as plant and seed identification courses, and is coaching the collegiate crops team with KSU. Like I mentioned before, that was previously led by Dr. Donnelly. Highly successful crop studying team and has won numerous national awards. And she is also the Wheat State Agronomy Club advisor, in addition to teaching. And she will also lead field-based crop science research programs, which she is currently being built. So with that, should be then turning it over to Rachel.
Okay, great. Thank you, Craig. See if I can get my screen shared here. All right. Well, thank you so much for having me. I'm very excited to be here. As Craig mentioned, of course, my talk today is about tillers. That's kind of what I've been talking about a lot for the past 4 years as a grad student. And today we're going to talk about the good, the bad, and the ugly of corn tillers. So we'll go ahead and get rolling here. I am not originally from Kansas. I'm from Southern Missouri, Hartville, Missouri, which, if you're unfamiliar with the great state of Missouri, is the lovely star that we see here on this map. But I grew up on my family's cow-calf operation in Southern Missouri, where we can raise a cow-calf pair on 1.6 acres. And so coming to Kansas, and especially Western Kansas, just the environment is much different. And getting to experience that has been a really cool part of moving here and doing this research. I got my bachelor's degree from Missouri State University in Springfield, and for the last 4.5 years I've been in Manhattan. During that time I finished my PhD here at K-State, and my dissertation, which is the 200-page book that they make you write before you can graduate, but I talked about the development and the drivers and the impacts that tillers have on corn yield stability in Kansas environments. And that's what we are going to dive into and talk about today. So the gist of what I'm going to be talking about is crop plasticity. And that's just a fancy word to say that crops have the ability to express different traits in different environments. So based on where you plant your crop, it's going to do different things. And that's not a new concept, right? So in plants, this plasticity ability, or the ability to adapt to different environments, is a natural characteristic that these grass plants have always had. So you either adapt to where you're planted or you die, right? And this trait, when we domesticated our crop species, we suppressed this ability, maybe potentially removed pieces of that through our domestication efforts. And currently, so the modern plants that we see of these crop species, the extent or the expression really changes based on which crop we're looking at. So I've got some examples here. We've got wheat, we've got green sorghum, and we've got dent corn, right? And if we look at sorghum and wheat, you may think in your management program to manage for tillers. If you're planting sorghum later, you might boost your population because you know it's not going to tiller as much, right? So that could be a component of our management strategies. In corn, you will never, ever, ever hear anyone say anything about a management plan that considers tillers in corn. And when we're talking about these crops and this plasticity ability, where they're showing these different characteristics in these different environments, we can talk about two different types of plasticity. So we have source plasticity, and source is just how that plant is accumulating energy. So in this case, that could be the leaf number, the size, the shape of those leaves, the branching, the architecture of our canopy. And then if we look at our sink, which is where that energy is going to, or where that energy is being used, we could think about our yield components. Roots are a sink, so the root structure, those different things. And when we're talking about tillering, corn tillers can be both of those things. So they increase our source, but they also can increase our sink. Historically, though, tillers in corn, which are the branches that we see from the base, right? They've been historically called suckers. And as a grad student, one of the things that I had to do was go through and comb through all the literature, anything that anyone's ever written about tillers, right? And it was really funny to me because some of the earliest publications that mentioned tillering in corn called them suckers. Like in the scientific publications, they were calling them suckers, which I thought was just really funny. But they've been masked over time, like we talked about with our domestication efforts, and just corn in general through our genetic selection, what we wanted in our modern corn hybrids. We just don't see tillers as much. They're still observed in some cases. If our plants have access to enough resources, they will put tillers on, and that's what we're going to talk about today. But generally, tillers are pretty unstudied, and the reports, the information we do have is not very clear, and there's not many strong conclusions that we can get from the information that we have available to us. One thing that I've noticed over my time as a grad student the past 4 years is that oddities pique interest. And corn tillers, when we're talking about research in corn, it's definitely an oddity. It's something that not many people are talking about, not many people are studying, but it's something that people see and everyone kind of has an opinion about. It's something that you hear talked about at the coffee shop on Saturday mornings, right? But these are just some pictures I've gotten from some farmers across the US. This is a photo from Canada, but some different people that are seeing tillers in environments beyond just Kansas. This particular picture from Iowa, Dusty, his planter had some problems and there were a lot of skips in parts of the field, and his corn plants were putting tillers on because they have those resources and they're going to use them. So what I'd like to do, just to make sure everyone is awake out there, in the chat, I'd like you to use the chat, please, and I'd like you to answer this question for me. So do you see tillers in your cornfields? A, never. B, occasionally, depends on the season. C, not really, only on the field edges, might see a couple on the edges. Or D, all the time. And if you answer yes, that you see tillers, what plant population do you typically target? So for example, if I see tillers all the time, I might put D, and then my plant population I typically target in those fields might be 17,000. So I'm going to give y'all just a quick minute to do that. I'm seeing some numbers starting to come in. Most of the ones that I'm getting are occasionally to all the time. A mix of populations. Looks like the lowest I've seen is 16 all the way up to 28. Yeah, good, great. So I'm going to keep watching that to see what's rolling in. But when we're talking about tillers, and of course the question today is, are they good? Are they bad? Or are they just flat ugly? Where do they fall in this range of options that we can talk about with our tillers? And to think about this a little bit, put this in some context, we're talking about plant density. That's a big factor when we're talking about tillering, which is why when I asked this question, I tacked on the plant density to that question. But if we look at this graph, so what we see here, we've got on the x-axis is our plant density in 1,000 plants per acre, and on the y-axis, we've got our grain yield in bushels per acre over time. The different line types and circles and triangles and squares we see here are hybrids from different eras. So if we look at these lines, for example, our circles down here, these are the populations or the optimum plant densities for hybrids that were from the 80s and 90s. We can see that they level off right here just below 30,000 as our optimum. If we look at some of the newer hybrids that we have on this graph, which would be from the 20-teens, we can see that optimum is marching further and further towards more and more plants per acre. So we can see this difference over time, and our preferences with how we're managing our corn is changing. We're moving towards higher plant populations, which is great if your environment can handle that. But if we're looking at the environments like probably what most of you all experience, that's not really feasible all the time. So this is taken from the Kansas Corn Production Handbook that K-State has put out, and you'll notice we've got our plant population, plants per acre. All of these ranges are within what you all were saying in the chat that you work with. But depending on where you are, that range can get quite large, and the optimum plant density in a given season can vary quite a bit. So if we could find a way to reduce our dependence on plant density in some of our environments where our optimum varies so much from season to season, that could be useful. There could be usefulness in that. And that's kind of where the tillering discussion kind of came into this. So to give you some background on our field study, the types of environments we were looking at and what we were testing. So the main factors we wanted to look at were plant density, hybrid, and tiller presence. Tiller presence and plant density were our two big ones that we were really wanting to look at. So our three plant densities were 10, 17, and 24,000 plants per acre. And we tested that in two pioneer hybrids. Both of them were prone to tillering. Would have liked to have some more genetics in there, but as a single grad student, you got to kind of draw a line somewhere, and 2 was our line. And then we were looking at tiller presence. So we either left the tillers intact for the whole season, or we removed them early in the season, and we targeted the V10 stage for that, and we removed them by hand. So there was no special way that we took the tillers off. We were out there on our hands and knees pulling tillers off the plants. But we chose V10 as kind of a compromise because we wanted to avoid V12 and later, because that's really when ear development is kicking in, and we wanted to have a minimal impact on those processes as the plant was developing those ears. But then we also didn't want to have to go back and take tillers off, because as you'll see on my map in just a minute, we were driving quite a ways across Kansas during the summers. And so tillers growing back that we'd have to go back later and take off wasn't really going to be an option for us. By V10, if you take those tillers off, they're not going to grow back. So that was kind of our thought process there. In total, we had field trials from 2019 to 2021. And you'll see here we had 17 sites where those were located across the state. I think these sites up here may be the only ones that are in the districts that we're talking about today, but we had a range of different environments that we were looking at across those 3 years. We took intensive biomass sampling, so really trying to understand how that plant was developing in 7 of our sites, which you see in purple on this map. And then The other thing that I definitely want to be sure to mention is these sites that you see in purple here were planted on the Corteva yield test sites, and they do have limited subsurface drip irrigation in those fields just to make sure that all their money that they invest on their research turns into something at the end of the season. So it wasn't full irrigation by any means, but it was enough to make sure we had a crop at the end. So did want to acknowledge that. So we're going to start with the good. Can tillers contribute anything positive? What good can they possibly provide? We're going to talk about some of the things that we found for that. So we published a couple things related to the yield response of corn to tillering. We have a research article that's out, and then we have a K-State bulletin, a 2-page summary of that research that's out also, if you'd like to check that out. This publication specifically is just for our first 2 years of data, but our third year agreed with our first 2 years of data as well. So the question, will tillers reduce yields? That's the big one, right? And in our case, that was not the case. So here we have 3 example sites that I pulled out. We could group all of our sites into 3 main categories. Either they didn't respond to tillers, they kind of responded to tillers, or they really responded to tillers. And so those are the 3 categories that I'm going to show here. And I see Craig has put that in the chat. Thanks, Craig. So we're going to look at our grain yield comparing with tillers intact and with tillers removed. So if we look at our Manhattan site, it was not a great field that we had in Manhattan in 2019, but our average yields, you can see we're in the 90s, and we have the same letter on top of both of those, which tells us that they are statistically the same. So it didn't make a difference in this case. If we look at Colby in 2020, we can see that we did a little better with our yields, right? But we still share a letter across these 2. So technically, the variation in the yields we saw in these 2 treatments, they're not different enough that we could say that there was any difference here. So these are statistically the same as well. If we look at Goodland, though, this is where it starts to get interesting. We had a couple sites that responses look like this. So with tillers intact, we broke 200. If we took tillers off, we didn't. And the letters are different, which tells us that statistically, we could say that these were different, that we did see a real difference here. So with tillers intact, we had higher yields in Goodland. Now, does the number of tillers that develop matter? And that's the question, right? Because some tillers will put on grain, some tillers won't. If we look at the Goodland response that we just saw, we had tillers that put grain on in that field. But how many tillers we have, does that matter? And the answer for that is that it depends on your environment, which is a very typical agronomist answer, I know. But it varies a lot by environment. And tillers can compensate for a loss of plants, so for a lower plant density, but only to a limited degree. And again, dependent on the environment. So this is kind of a rough figure, but we're going to get through it. So hang on. So on the bottom, we've got an increasing plant population that we're going to look at for each of these 3 sites, the ones we just looked at, those 3 categories we saw earlier. Our plant population, again, ranged from 10 to 24,000. So that's what we're going to be seeing there. And then our tiller population, so the number of tillers we saw in these plots, we've got a range from 0, where we took those tillers off, to 33,000 per acre that we saw with tillers in these plots. So if we look at Manhattan, what you'll notice is the curve of the lines, right? So the darker purples are going to be our lower yields. These lines are the contours of where our model would predict how much yield we would see from a combination of plant population and tiller population. So in this case, you'll notice as we go along the x-axis and along the y-axis, there's not really much of a change that's happening, especially with our tillers. There wasn't any response to that. If we look at something like Colby, okay, what we start to see developing is more of a gradient on both of these axes, right? So in this case, the lighter colors are higher yield, colors are lower yield. So as we go from a lower to higher plant population, we're adding yield. Our model is predicting that we would see more yield with more plants, but it's also telling us that at low populations, we could see that gradient strongly with increased tillers developing as well. In Goodland, Now this is where it gets interesting, right? So we can see that we've got an increasing plant population, a really strong gradient here, and just as strong of a gradient with our tiller population. So what that means is if we look at, let's say, a plant population right around here, which would be, I don't know, we'll say that that's 20, just shy of 20, maybe 18,000 plants per acre with no tillers. So that'd stick us right here, would predict to yield the same as a 10,000 plants per acre with, with around 20,000 tillers. So that compensation that I talked about tillers can compensate for lost plants, that's what we're seeing with the curves in these graphs, is it's showing us how the model would predict that happening. So this is an example of that plant in Goodland that I was telling you about. So we've got a main stalk with 2 ears, primary and secondary, and then we've got 2 tillers, and each of those tillers has, you know, an ear on it, right? So that doesn't always happen. It can. We're going to talk about that in just a minute. But tiller presence didn't reduce yield in any of our evaluated environments or plant densities. That's a key takeaway there. They could produce ears and harvestable grain like we see in this picture. And in favorable scenarios, so in certain environments, corn tillers had the ability to compensate for plant density to a point, right? Now, the real question, do corn tillers reduce whole plant reproductive efficiency? So those tillers are putting on a lot of biomass. There's a lot of extra shoots that are developing here. So what is that doing for something like our harvest index? Now, in our case, looking at the tiller development, the tiller presence impact on full harvest index, we did not see any relationship between those two things. So if we look at this graph, we're going to have harvest index on our y-axis and tiller biomass on our x-axis. So we're going from 0 to 5.3 tons of dry shoot biomass per acre for our tillers that developed. And what we see here is that these points, looking at the harvest index from these plots compared with the tiller biomass that they put on, our line is flat right at 0.5. So what that tells us is it didn't matter how many tillers we had, how big those tillers were, how much biomass those tillers were putting on. We did not see any direct relationship between that tiller biomass and the harvest index of those plants. So when we start talking about biomass, carbohydrates is where we need to go, because the corn stem is essentially a storage organ. And what happens is as that corn plant is growing and developing, it stores sugars in its stems, and that sugar, that energy, is then moved to the ears to a certain degree during grain filling. And so if we have the ability to measure the carbohydrates in our stems and how that's changing over time, it can be an indication of the energy needs and the internal balance and the energy in that plant, of what that energy is being used for. Is it going to the grain? You know, what's the plant using that for? And WSC is an abbreviation I'm going to use in the next couple slides, and that's just water soluble carbohydrates, which is what we were able to measure to look at these relationships in the plant of where this energy is being used. And so I've got another question for you that we're going to stick in here in the chat real fast. So this picture, I'm holding 3 stems, 3 stalks, right? I would like you to tell me which stem of these 3 belongs to a tiller of these 3 that I'm holding. So which stem or stems belong to a tiller and which one doesn't? So ABC, which one is a tiller stem? A is a tiller stem. I'm seeing some guesses coming in. A's, B's. It's kind of hard. When you look at these pictures, and even when we were taking samples out in the field, we cut the plants and we'd rubber band them together so we knew which tillers came from which plants. But we'd get back to the shop to do our processing, and it'd be kind of hard to tell which one was the tiller and which one wasn't, because the stalks were about the same size. They were about the same height. So I was very impressed with the size that we could get with these. So it looks like everyone's saying A. Actually, both A and B are from the tiller. C is a main stalk. So those tillers can be about the same size as our main stem, which in this case was C was our main stem, A and B were our tillers. So I thought that was pretty cool. When we're talking about carbohydrates, we're talking about the reserves that these plants have in their stems to use later. And what we're going to look at here is a comparison of plants from our 3 different plant densities that we were looking at. So 10, 17, and 24,000 plants per acre. We've got growing degree days on the x-axis. So we're looking at dynamics throughout the season. So we go from planting to harvest. We're looking at how that's changing over time. On the Y, we've got the water-soluble carbohydrates in grams per plant. So this is the total content, the carbohydrate content in those plants over time. You're going to see 2 lines on this graph. So here we've got our solid color gray area here. That is our water-soluble carbohydrates in our plants without tillers on them. And then our dashed dark blue line is going to be our carbohydrate content in our plants with tillers intact. So you can see that we've got a difference, right? Our ranges, our variability in those, we're getting some separation in our 10,000 plants per acre. If we look at our 17 and 24, what you'll notice that there's quite a bit of overlap. Now, the key thing I want you to see here is that we can draw a line right across the top here, and there's a pretty strict cap to the amount of carbohydrates that a plant can store. So it doesn't matter if it was put at a 10,000 to 17,000 or 24,000 density, the per plant ability to store carbohydrates, to store this energy, was pretty capped. The only difference that we see is our 10,000 plants per acre with tillers intact. And in that case, the energy that was left in that stem at the end of the season was almost double what it was per plant if we didn't have those tillers. So what this tells us is that tillers were able to increase the water-soluble carbohydrate reserves in these plants by adding these extra stems. So most of the biomass, the shoot biomass that was coming from these tillers, was put into stems, and so the plant could essentially use this as a bank for carbohydrates. And that ability to buffer the water-soluble carbohydrates, so to put more into the plant than just what the main stem alone can handle, is that this can increase standability. So later in the season, if that plant is having to pull a lot of reserves from the main stem, we can see lodging if those stems are weakened from moving that carbohydrate around. But if the tillers are an extra source of that, then we're not relying as heavily on our main stem for that. Stabilized kernel weights is also part of that, because if later in the season our plant is stressed, it's water stress or heat or whatever your problem might be, and it's not able to photosynthesize as well, if we have extra storage, we're able to remobilize that for our kernel weights, which is a big part of the reason we think that we saw those stabilized yields even in areas with tillers that maybe didn't see much of a yield boost, but they weren't hurt because tillers were able to support the carbohydrate reserves in this way. And the ability to use that energy, so yes, we found evidence that the water-soluble carbohydrates, that this energy source was moving from the tillers to the main ears. There was a study done in the 1970s with labeled carbon that also was able to trace the movement of carbon from a tiller leaf to the ear on a main stem to prove that that could happen. And then there was also a study done in the 80s with phosphorus to show that phosphorus could move from tiller tissue to main stem tissue as well. So it's a bank potentially for energy, for carbohydrates, but it's also been proven as a source of mobile nutrients in the plant that can be moved from the tiller to that main shoot as well. So the bad. So that was the good. So now we're going to get to some of the bad stuff. Key questions that we have with this is, okay, that's great, but what about all this extra leaf area? Because if you've got more leaf area, you've got more areas where that plants can lose water, right? And then how on earth can you predict how these tillers are going to do? Are they going to yield anything? How are they going to develop? Are they going to be there at all? And so those are questions we're going to go through. So some people, if you saw tillers like this in your field, you might think, oh, that you've got happy plants. And other people might see this and think that it's just an absolute disaster, right? So there's some very legitimate concerns with tillers that I wanted to talk about. And we're going to go through those. So yield is a big one. How's it going to impact yield? Water is a big one. The nutrient use, so what are these nutrients being used for something that's good, that's going to be usable, that I can harvest later? The harvest index, water, right? Tassel ears, we'll talk about those in a minute. But then we've got predictability, which is how are those tillers going to perform? And then water, of course, right? And you may have noticed that we've got a theme going here. But yield, so talking about yield specifically and the concern with yield. So our studies show that there were no reductions in yield just because tillers were present. Now, that doesn't mean that that could never happen, right? That doesn't mean that yield reductions are impossible. But the fact that we had 17 sites that did not show any indication of a reduction in yield gives a very strong case that we can't assume that just because we see a tiller, our yields are going to be penalized. So that assumption, that sucker assumption, doesn't hold. And so the basis of this would be if you see tillers, don't panic, right? Yield-wise. We're talking about harvest index and nutrient use. So our studies again showed no significant impact on harvest index just because we had tillers there. And again, just like we talked about with yield, that may not always be the case, but our study gives a pretty strong case that we can't assume this to be true, right? It also agrees with previous research that those carbohydrates, like we talked about, and the nutrients can move from those tillers to the main stock. So there's some opportunities there. So again, you see tillers, don't panic. Okay. Water, right? Big concern is water. And I'm sure that I don't have to tell anyone in this webinar about how important water is for corn, for farming in this part of the world. To give some context for our sites, the seasons that we had range from 13 to 22 inches of water that these fields got, whether through precipitation or that limited irrigation I was talking about. And our yields ranged from just shy of 100 to over 200 bushels per acre. So we had a range of environments. Of course, if we would have done this study in 2022, we may have seen some different results. There have been some field studies in Argentina and other parts of the world that were looking at tillering in a similar way, and they found in field studies that there were not significant differences in yield. In modeling studies, like computer models, they did find a couple situations where with low enough water, there was a penalty with tillering. But the question is, where is that line? Where does that break point happen? Where does that change? And that's something that we don't really know. So the idea behind this is if you have more tillers, you have more leaf area, and that means more transpiration, which means water moving through those plants is being lost into the air from the soil. And a lot of times, what I've heard people talk about, and it's a good strategy for this maybe, but the alternative planting geometry, so skipped row or clump planting, different ways that we can plant corn to increase the humidity in that canopy. So those leaves are closer together, we're closing that canopy faster in certain parts of the field, but then also we're extending soil water because the roots of these plants have to grow to these inner rows to explore that water. Each plant has more of a field area that it can explore. And then also if we have clump plants, they're shading each other out, so we see fewer tillers. So there's a lot of things at work here when we're talking about alternative planting geometry. Tillering is a part of that, but there's other things that play into that too, that can help with water use too. So the question going through all of this is, at what point do tillers hurt more than they help? And I was talking with a guy at a corn school this past year actually, and it's the question that he had for me is, what is the point where tillers are hurting, or is the plant just dead at that point? And how do those two mix, and where's the line? And that's something that I don't have a good answer for. You never hope for a dry year, but with research, if we would have had plots in 2022, we may have been able to get more of an answer in that regard. But water is something that I don't have a clear answer for. The other part of this was the tassel ears and the predictability. So can we predict tassel ears? Can we prevent tassel ears? We're going to talk about those in just a minute, but how do you predict how well a tiller is going to yield? How can you predict that? Or if you're going to have one at all? And one thing that I noticed when we're in the plots, when we're doing these types of things, is you'll have a plant that has 3 tillers on it, and those tillers put ears on and it's great. And then the plant next to it doesn't have any tillers at all. And then the next plant maybe has 1 tiller and it puts on a tassel ear. And it's like, it's so interesting to research. And as a grad student who had to take data on that, just seeing the variability in that, I think predicting this, it's possible. We have put some models together that we're working on, some other projects with this, but we're working on it. So hope to have some better answers for that in the future. All right, so we talked about the good, the bad. Now let's talk about the ugly. And when I think of ugly related to tillers, tassel ears are definitely what comes to mind. Those are ugly. Okay, so we talked about our yield increases, and those were coming from our kernel numbers. So our high kernel numbers were most closely associated with those yield increases. And to get those more ears, we've got more kernels, right? And ear type is important to making sure that we're getting those kernels. So if we look at ear type, we've got a couple different things that can happen with our tillers. We've got primary ears, which is on the main stalk, the top ear on that main stalk, right? These are our yield drivers. This is why plant density is key, because we need to get as many primary ears on there as possible. Secondary ears, we're looking at the next lowest ear on that stalk, and this is a form of plasticity. So in a good year, if you've got a prolific hybrid, that would be a way that those plants can express some plasticity and try to get some more yield. Tiller axillary ears or typical ears are the typical looking ears that we see that develop on tiller stems. So this would be an example of a tiller typical ear. These are harvestable. Usually our tillers are about, I wouldn't say more than a week behind in development. So the moisture percentage at harvest and the pollination of these is typically pretty good. The other ear type that we can see, though, is our tiller apical ears or our tiller tassel ears. And these are atypical, they're ugly, and they're confused mixes of ear and tassel. So if you see in this diagram here, we've got some kernels, we've got some tassel pieces, but they're not harvestable because they don't have a husk that develops around that ear. And so what happens is bugs and birds and weather and smut and everything else you could think of gets, you know, all you can eat buffet on these kernels that are available at the top of this plant. And basically what happens when tassel ears are developing is corn is unique because it has male flowers at the top, female flowers in the ears. But what happens sometimes is the process gets a little bit disrupted because when these flowers are first developing, we have male and female in all of the flowers. The female flowers in the tassel are selectively aborted and we're left with only male flowers. In the ears, all of the male flowers are aborted and we're left with only female flowers. And what happens with a tassel ear is that process just gets a little messed up and we end up with some female and some male mixed in our tassels. And we don't like that because, like I said, we've got birds and bugs and smut and everything else that has access to those kernels, and we're not going to get anything good out of them. Here we have from a tiller, this is a tassel ear as it was developing on a tiller. You can see those silks starting to come on. We've got those kernels developing, but then we've got tassel pieces in there too. This is a typical ear and a typical tassel also from a tiller. So the interesting thing to me that was very surprising is we were looking at how this development, these tassel ears developing, how that impacted yields. If we could specifically look at plants that had tassel ears that developed and plants that didn't have tassel ears develop. And we did not see a yield penalty when we had tassel ears present. We definitely didn't see a yield boost, but we didn't see a penalty from those tassel ears. So we're going to look at our primary, our secondary, and our tiller axillary ears to see how that impacted our yields that we were seeing. We have low, medium, and high yielding environments, and we're going to look at the yield contributions of each of these ear types. So if we look at our low, you'll see that our model couldn't really tell the difference between the 3 ear types. And this makes sense because adding more plants, so increasing our density or increasing our primary ear number, didn't really help our yields in our low yielding environments because we were maxed out at a low plant density. So we didn't see any clear distinction between those ear types there. If we're looking at our mid yield environment, what you'll see is that primary ear starts to pull away as what the model can say, okay, most of your yield is coming from your primary ears. And that makes sense because we're able to add more plants to increase our plant population, and we're seeing that here. But our secondary and our tiller axillary ears, in this case, the model couldn't really tell the difference between the two. In our high yielding environments, we see that our primary ear is pulling away as more and more important. And as we know with corn that in high yielding environments, plant density is really, really important. But what I want you to notice is that our secondary and our tiller axillary ears, our tiller ears, our model actually was able to tell us that there was more yield. If you could get more yield from tiller axillary ears, more grain coming from tiller axillary ears, that could out yield a secondary ear on your main stock, which I think is wild. But if we think about why that would be, so if we think about our primary and secondary ears, tiller ears have less direct competition than secondary ears would. And the reason for that is our primary ear on a corn plant, the photosynthesizing leaves are mostly up here and our sugars are going down. And as those sugars are going down, the first stop is the primary ear. So that ear gets preference over the secondary ear. So it's like the youngest child, first child. I'm an oldest child, so I can say this, but the primary ear is actually the last ear on the corn plant to develop. So it's the youngest child and it gets preference, right? As we know, that's how it always works. But if we go to our tiller, we can see the ear on our tiller. And this doesn't have any other ears that are competing for the sugars that our leaves at the top of this tiller are giving. So this tiller axillary ear gets all of the resources that this tiller is giving from its leaves. This secondary ear has to compete. So if you think about it, that makes sense. And essentially what we're doing by adding these tillers, by adding these shoots, is we're mimicking more of a plant population. So we're mimicking that we're adding to our plant population. So your to-go box for today. I've got 3 key things that we've talked about that you can take with you. So key points first would be that tillers did not reduce corn yields in any of our trials. We did not see that in any of our 17 locations across the 3 years that we looked at. So it doesn't mean it can't happen, but we made a very strong case, like we talked about, that we can't assume that we're going to see yield reductions. We cannot make that assumption. The environment impacted the number of tillers we saw and how those tillers performed. So that could be the amount of yield that they were contributing, whether or not we had tassel ears or not. There's a lot of things that we can look at with environment to try to help us with that. That's a work in progress. But then we also saw that tillers increase those energy reserves in the corn plant and that those reserves can move throughout the plant. So if you remember the carbohydrate curves that we looked at, that extra bank of carbohydrates of energy that tillers had were able to contribute to that corn plant. We could see that in our lowest plant population when we had those tillers developing. And with that, I think I have reached my allotted time for talking. Thank you so much for having me. If you have questions, I would love to take them now. I've also got my contact information on here. My Twitter handle is on there. And would love to connect with any of you if you have any questions. Thanks.
Rachel, this is Sandra Wick. It looks like there is one question that popped in and you probably see that in the chat.
Oh, yep, I see it now from Jay. So it says, did you see pollination variability with tillers? Is that a concern in a high tiller environment, good or bad? So pollination variability is a really interesting question. And I'm going to answer this with an example that we saw in Garden City. So the pollen on a corn plant, if you think about the main stalk, it drops pollen for about 3 to 5 days usually. So you have that 3 to 5 day window for pollination. And if you have really hot temperatures during that time, you're kind of in trouble as far as your pollination goes. We had tillers in Garden City that pollinated about 3 to 5 days later than our main stem did. I think that was in 2020 that we saw that. But because they pollinated 3 days later, our ears on our main stalks looked pathetic because the pollen was sterile and it didn't fertilize our kernels. But our tillers had much nicer ears just because they missed that really hot time when they were pollinating. So there is some variability in the timing of pollination. That's not always a bad thing. In our case, we've never seen that difference in timing to be a reason that we have poor pollination in our tiller ears or anything like that. And then if you have pollination variability, you're also going to have a difference in maturity by just a little bit. A question I get quite a bit is the moisture of those tiller ears compared to the ears on the main stalk. And my answer for that would be that the variability between those tiller ears and those main ears on the same plant, there was less of a difference between those ears than between main ears that you would harvest from different parts of your field. So I don't think that that's something to be concerned about. Let's see, I see another question that popped in. Fred, thanks for the question. Any thoughts how tillers might affect silage production? So that's actually an interesting question. And there are specific hybrids of corn that are planted or bred for silage production. And in those cases, you want tillers in those because you're getting more biomass in that production system. And there's been, trying to remember, there was a study that was done in Canada looking at the impact of tillering on silage production actually, and how the number of tillers in those silage hybrids was impacted by plant density. Generally, it was favorable if I'm remembering correctly, but that extra biomass could be good if you're using it for something like silage. Yes. Storing water-soluble carbohydrates might affect nutritional quality. So yeah, if you've got, and that runs into how big are your tillers getting, right? And are they putting ears on? If you've got all this extra starch in your stems, that would be something to balance out with the nutritional quality. I don't know, Fred, if that was meant for silage, or if you're talking about grain quality. Grain quality isn't something that we looked at directly. I wouldn't expect there to be much of a difference in that either. Did we have any other questions? Did you all see any other questions?
I didn't. I was just looking through. Oh,
Just
go
had
ahead.
one quick one from YouTube. With the tiller trials you did, what type of irrigation did you use, and did that make any difference?
Yeah, so the slide that I showed, so in some of our locations that were on those yield test sites with Corteva, they had subsurface limited irrigation in those sites, and it was just enough to make sure that we had a crop at the end of the season. They weren't fully irrigating that, like deficit irrigation by any means. We did have in Colby, both of our sites, or I guess we had 4 years of sites in Colby. Those were not irrigated, and the ones in Manhattan, of course, weren't irrigated either, but that environment, when you compare it to a Western Kansas environment, it may as well be irrigated. So we had limited irrigation in some sites, dry land in some sites. We tried to get a range of environments that we were looking at. If we would have included more dry land, my guess would be that we would have seen less sites that had a yield boost with tillers, and we would have seen more of the like no impact of tillers in those sites.
Awesome. Well, thank you, Rachel, for spending time with us today. It's always fun to listen to your presentations. So we also wanted to remind you, if you are on here for CCA credits, please make sure you email Jeannie Polk Jones, and her email is on the the screen here, or you can scan your QR code, but make sure you include your name and your CCA number. She'll make sure that you get those CCA credits that you worked so hard for. And then we also have a evaluation that we would like for you guys to fill out for us. This helps us with future planning for next year's or the years after CropTalk series that we host. It also helps us with programming throughout the year. So if you could take a quick minute, it's like 3 to 5 questions. You can follow the link on the screen. It's also been posted in your chat box, or you can scan the QR code and it'll take you directly to the website. Craig, thank you. And once again, thank you for joining us for our 2023 KSU CropTalk webinar series. We've had a blast putting this on for you guys. And so everyone that joins, it's a special treat to see everyone. Recordings and handouts are posted on the KSU Northwest Agronomy website, or some of your local units will have it on their websites as well. The link is listed below. It is also hyperlinked in the chat box for easy access. And then just always make sure if you have any questions, you can always reach out to any of your county or district officers, and they would be more than happy to connect you to whoever can get you the most information the quickest. So with that, if anyone else has any other questions or they need anything else, just feel free to ask.